So let's welcome Dr. Manish Kumar to lead the virtual session on advanced analytical techniques, non-targeted analysis, and suspect screening.
So much eminence business media to giving me opportunity to present my presentation and discussing about the NTA and SSA enhanced impurity detections. So this is a very important topic. So I will go through the presentations. This is the overview, and in the overview, it's very clearly defined that identify the unknown impurity. Basically, this is the NTA during the drug development and post-approval of drug product. As per ICH-Q3A, it's a required unknown impurity present in the drug substance with the label of 0.1% must be characterized. So it means the regulatory body, regulatory expectations and the quality expectations is clearly defined that the known impurity should be well characterized and well within the defined label. And this NTA, basically unknown impurity, may be coming from the different sources. It may be the environmental contamination, extraneous peak, unknown peak may be coming from during product development. It may be the degradant, genotoxic, hydrosamine impurity, cross contamination, or any other excipient which we are using in the product development. And maybe within the matrix, it may form the adduct also from the and this NTA may come from the packaging material. So you might be heard about the leachability and extractability. That's the reason we are performing the LNE study for the parental product and ophthalmic and all. So that impurity may be coming from these, environmental and packaging material as well. And these NTA can be, identified from the high-end instrument technique like mass spectroscopy, especially from LCMS and GCMS, because the sensitivity is very, very high in this instrument. That's the reason we are going to use these high-end instrument to identify this unknown impurity. Going to the next slide. The next slide is defining that what is actually NTA. This NTA is nothing but the analytical approach using the detect and identify the broad range of chemical substance. Basically, the chemical substance would be the organic impurity, inorganic impurity, any kind of impurity without prior knowledge of the specific substance or present in the matrix on the product. The source which we have already discussed during post degradation study, during method validation, during product development, method development, during stability studies and all, complete during R&D stage, this unknown impurity or NTA may come. To identify this unknown impurity or degradant product, the conventional method like HPLC usually use most of the cases. In an HPLC, this unknown impurity can be checked with the RT of unknown impurity or NTA substance during product method development activity. And this high-end instrument like LCMS, GCMS, and HRMS technique will support to identify this NTA. So NTA, as we discussed, that NTA maybe from the chemical space is coming, and known compound would be the target analyte, and suspecting screening analyte would be the known and unknown, because the known and unknown, if the unknown impurities are there, but from the literature support or from the library of the mass, software, that will become a SSA. And if it is not available any information related to this particular impurity, then that would be called as the NTA. And coming to the analytical approach, definitely when we are developing any kind of method, we have to have a target for each and every impurity. That impurity may be the process, degradant, or any kind of matrix from the excipients, placebo. That all impurities are defining. And apart from this unknown impurity, maybe the certain limit would be there as per ICH or MDD, target limit. So these are like, chemical space and analytical approach we have to adopt for identify this NTA. And this basically NTA is a, defining in the life cycle management of the product. when we are developing any kind of method during product development at R&D stage, we have to have some target for known impurity as well as the unknown impurity within the defined limit as per ICH, as per regulatory body expectations. So once we define the specification for the particular impurity, then we are going to do the, post approval for the product. If any unknown impurity may coming in the during analysis of the batch analysis, commercial analysis, stability analysis, then we have to go for the complete investigation on the hypothesis and using the high-end instrument like mass spectroscopy, and we have to confirm that particular impurity. If these impurities are coming from the matrix, from the environment, then we have to confirm. And during confirmation, we have to inform to the agency and get the permission whether it is safe with respect to the safety norms. And accordingly, we need to include the specification and continue the commercialization complying the defined specification. So the next slide is showing the suspect screening analysis. As I said that uh, suspect screening analysis in the sense any target impurity when we are developing a method, then we know that what is the target impurity, and during the method development activity or during analysis, we may come to know that these are the impurities, unknown impurities are coming apart from known or unknown. Then we have to have some database, the library, and that library can help us to determine these impurities may be matching with this particular mass. Then we can go and take the support structure from the library and make it like SSA. And once the SSA is confirming, then we have to identify and characterize and validate this particular impurity if it is going to be make it known. So this is SSA is clearly suspecting screening analysis during method development activity with the lower limit of the impurity, the response and the unknown impurity limit would be very sensitive. Technique can be used like mass. This slide is clearly defined that, as I said, this target screening would be the known impurity, whether like, process impurity, degradant impurity, any category of organic, inorganic, and genotoxic impurities, or maybe nitrosamine impurity would be the target screening. If any unknown impurities are coming, whether this known or unknown, it means the database would be available, then it may come into the category of suspect screening. And if it is not available any database, purely it is a known impurity, then we have to use the mass technique and high-end instrument technique to make it known and identify the actual cause of the impurity from where it is coming, and use the proper safety norms to make it known. Now coming to the regulatory integration, that what regulatory guidelines are saying about this NTA and SSA. The SSA is very clearly defined that the product comprehensive guideline for classification of identification of impurity and the unknown impurity basically in the product and chemical synthesis and drug substance and drug product. And as per the classification, this organic impurity, inorganic impurity, residual solvent, and any other nitrosamine impurity may be classified as per the safety norms. And as per ICH, the threshold of the impurity should be reported. And the qualification, if the impurities are well within the as per ICH guideline, otherwise we have to do the toxicity and prove it that it would be that impurity would be safe or similar to the innovator product. And as per ICHQ9 is clearly defined that synthetic approach for quality risk management during the life cycle of the product development or product, the decision tree should be defined. Whether this impurities is clearly characterized and defined and better to use for the resources and improve compliance and regulatory expectation and enhance the product quality and patient safety. As I said, the toxicity is the uh, final database which is going to help us to uh, identify this unknown impurity. And in the complete analysis cycle, we have to follow the ALCO plus principle. which is giving a complete confidence for the product quality, patient safety, and the regulatory compliance environment for this particular analysis. And these are the high-end instrument we are using for the analysis. This nowadays in the industry, like triple quad MSMS systems are using for identify this NTA, HRMS, time of flight, orbited are using the mass accuracy within the ppm, and even then ppb level also is very, very important, especially in nitrosamine impurity and genotoxic impurity. We have to go with the lower level, trace level, and ensure that impurity should be compliant as per the regulatory expectations. And in these high-end instrument, the ionization source are available, and we have to use the ESI mode as well as the APCI mode for depend upon the nature of the impurity and nature of the compound. And these mass spectroscopies are coupled with the liquid chromatography as well as the gas chromatography, because usually in the analytical or quality control, we use the liquid chromatography as well as the gas chromatography for the analysis purpose. Now, coming to the workflow of this mass spectroscopy using, and how we are going to do the unknown impurity identification. So, if any unknown impurities are coming up, then we have to develop the mass compatible method, because the liquid chromatography method may not be suitable for the mass, because the mass is a highly sensitive technique, and this buffer, basically the volatile buffer, is going to be used for this mass spectroscopy, and we have to develop the method. And once the method has been developed, then accordingly dilution, filtration, and solid phase extraction need to be done. And the data acquisition and processing, the ASI, APCI mode, full scan, we have to do it, and according to the impurity, according to the runtime, we have to develop the method, and using the different you know software, that impurity assessment and impurity identification and RT confirmation is going to be helped by these software. After generating of the data analysis data like mass and so many data are available, then these software is going to help us to trending the analysis, because the chromometrics, the tool for extracting information from multivariance chemical data using the statistical and mathematics, and the principal component analysis, clustering of the visualization trend, and these data uh, need to be reviewed and evaluated properly, and so these software is going to help us to make it the trend. And if we have a multiple batch data of stability, then the smart stability and NovaTrack software is going to help us to do the proper extrapolation and the particular impurity trending. And this is very, very important. Then if we are going to do the multiple batch manufacturing and analysis during the year or during the multiple years, we have to do the proper risk assessment for all these data, whatever the data we have generated so far, and sending the batches to the in the market. So the complete data assessment need to be done as per the quality system to assess the impurity risk. If these impurities are unknown impurities are coming in this particular batches, then we have to assess the complete past data as well as to extrapolate the upcoming data as well. And this is the interpretation. As I said, this unknown impurity or NTA may come from the different sources. Basically, the NTA may be the part of the within the product matrix, or it may form during the product development. It may form during the degradation, or it may form from the within the matrix. If any excipients or any alternate vendor change may be there, then it may coming from the within the raw material source within the matrix, or it may coming from the environmental contamination. Environmental contamination may be from the cross contamination within the quality control lab. If any analysis may take place, the glassware may also may contaminated this unknown impurity. So excipient, placebo, packaging material also very, very important for generation this unknown impurity. So during hypothesis, during investigation, and during the complete analysis, or investigation has to be done in the quality control department if any unknown impurities are going to become. So as per the EDQMA guideline and FDA guideline clearly defined that we have to do the proper investigation to identify this root cause of this unknown impurity with the phase 1, phase 2 and phase 3 analysis. And this is the investigation process flow is clearly defined that what would be the suspected, unknown impurities and from where it is going to be come. So we have to define the fishbone diagram. We have to define the, source of this impurity. What would be the, source of this impurity? And during the product analysis, these are the, material is going to be used. So each and every material, each and every source we have to use and check whether these impurities are coming from this particular source or not. So maybe the raw material would be the change or intermediate would be playing a role to carry forward this unknown impurity. Degradation may take place for this particular impurity or any packaging material also any adduct formation may happen due to change of any raw material or API source and all. So all these things we have to do during investigations. And the contamination also very important. Like as I said that glassware during analysis, any adjacent person is doing analysis, if any glassware has been contaminated or any change, something happened. So in during phase 1 analysis, phase 2 analysis, these all investigation is going to be supported and these are the probable, source of contamination coming for this unknown impurity. So we have to do the proper, root cause investigation during hypothesis, during investigation in the quality control department, or for identify this unknown impurity form it is coming up. And once we can gone through the complete hypothesis and phase 1 phase 2 investigation and we could not able to succeed that unknown impurity source from it where it is coming up, then we have to use the high end instrument like mass spectroscopy and all. So once we use the mass spectroscopy, then we may come to know that what is the mass of this particular impurity. That mass is going to help us to identify the probable root cause for this particular impurity. And if we come to know that this may be the probable root cause or probable structure or prediction, we have to done it. And probable structure will help us to identify or evaluate this particular impurity. This particular impurity or organic impurity, inorganic impurity, or any nitrosamine impurity would be the category of which category. So we have to run the in silico module and all. Then we have to identify whether these impurities are coming under genotoxic category or whether it is organic impurity. Then we have to go for complete synthesis of this impurity and do the characterization properly. And that particular impurity, once we isolate it, synthesize this impurity, and we have to confirm in the particular HPLC analysis, confirming the RT. And once we confirm the RT, then we have to do the proper validation of this RS method, spiking this impurity, and check whether this impurities are impacting the existing analysis, what would be the RF, what would be the RT, and if RF is within the 80 to 120 range, then analysis data need to be evaluated properly and check the safety risk of this same. If the data coming within the MDD within the ICH guideline specification limit, then we can go and revise the specification, generating the data for inhibitor as well, and inform to the agency. If the unknown impurity data after applying the RRF is going beyond the ICH limit, then we have to go for the toxicological study. In the toxicological study, we need to perform the different animal, maybe the rat and mice and all. And once the toxicological study has been completed, defining the percentage of the impurity limit need to be toxicological study. Once there is no adverse reaction observed during toxicological study, then we have to do the proper validation, applying the higher percentage of the limit in the particular RS. And once the validation completed, toxicology report is available, then we have to update the specification and inform to the regulatory agency to taking a permission for higher limit of this particular impurity. So this is the whole process of investigation for NTA as well as the unknown impurity. So as I said that once we identify the impurity, unknown impurity become a known impurity, from like unknown impurity to SSA to become a target analyte, and the target analyte we have to validate the method using the ICH guideline as well as the USP. And these parameters going to help us to validate the method, whatever the existing method we are using, where the unknown impurities are coming up. Once it is make it known, then we have to perform the complete validation using these all parameters like LOD, LOQ need to be established for this particular impurity, solution stability, specificity, precisions, and robustness. And apart from that, method transfer is also very important. If we are going to use this method in maybe CMO or any quality control department, if you are going to do the validation at site, that is also okay. If it is doing the validation at R&D, then we have to ensure to do the method transfer and ensure that this method or this particular method, whatever the validation has been done, it is going to be accurate and robust prior to batch analysis for this particular impurity. So the method transfer is also very important as per USP general chapter. Now coming to the case study, I have described a couple of case study to understand in a better way what are the in pharma industries we always facing this kind of challenge for unknown impurity during post approval commercialization manufacturing and during analysis at quality control. And this slide is clearly indicated that during batch analysis of topotecan drug product during post degradation, we come to know that unknown impurities are coming up. That unknown impurity is 1.06 RT, and this unknown impurities are coming uh, around 7% in post degradation. So we have to make it known. We have to identify okay, what would be the impurity. So we completed this mass spectroscopy analysis in the ESI plus and ESI minus mode. We come to know that the mass would be 345, and this mass.
I am so sorry to cut you there, Manish. We've got a message, WhatsApp from one of the delegates that it's a complex information as well. So if we can slow down on the speed of the slides,
Okay.
so this feedback we got. So this is why I thought that I'll just highlight it. You can take your time. We want to everyone to make sure that they are able to understand everything. So you can slow down on the slide speed.
Great, great. Thank you so much, Meet,
You're
to
most
inform
welcome,
me.
Manish.
So I need to check that, yes, I'm not going to be fast because we have
Yes.
a sufficient time. Okay.
If I have
So
to
can
give you
I,
extra 10 minutes as well, that is absolutely okay. We can do that. We do have buffer of time in hand. So don't worry, you don't have to rush the slides.
okay, great. So whether we need to go any backslide or I need to continue with the same?
I will check that, and if we do, then I will intervene and let you know.
Okay, perfect. So,
Yeah.
as I said, in the case study during, product development in the post degradation, we come to know that there's the unknown impurities, and this unknown impurities during degradation, we found that yes, this would be the probable, impurity during stability. It can form. So we have to identify. We have to ensure that im- impurity should be, characterized impurity should be identified during development stage. So we check the mass and taking a help from mass spectroscopy LCMS, and we develop the method compatible develop method with the mass spectroscopy, and we identify the mass is 345. Once we identify the mass of 345, then we go on for prediction of this structure. Okay, what would be the the structure of this particular impurity, whether we can make it known or we have to check during stability, these impurities are not going to be increased. So we predict the structure, ki what would be the, this particular impurity. So we are using a topotecan API, okay, in the product, in the drug product, in the tablet, and topotecan is a particular structure, and during acid and base post degradation, environmental like acidic and base degradation, this acid impurity may form because the ammonia molecule is going to be removed and this acid impurity may form. And this acid impurity of tofacitinib in presence of methanol and hydrolysis happened, then methyl ester impurity is going to be formed. So this is the production of this particular impurity with the help of 345 mass. Then we gone for synthesis and we synthesize this impurity and we confirm it spiking in the HPLC analysis, and we confirm the RT is matching. And again we have performed the mass of this LC, and once we got to know that yes, this is the only impurities are going to be degraded in the post degradation, then and we perform the validation activity, and this unknown impurity make it known, and this methyl ester would be the part of the specification. Yeah, coming to the case study 2, there is another product in cephalexin, where the cephalexin oral suspension, powder for suspension, we usually use the sucrose, the maximum part of sucrose in the formulation, and we come to know that there is a unknown impurities are observed at around 46-45 minutes, and this unknown impurity during stability is going to be increased. So we need to identify what would be this impurity, because the usually unknown impurity limit is the zero point 0.2 as per MDT, and the impurity limit is observed more than 0.2%, it is the 0.27%. So we have to identify what kind of impurities are there. After complete investigation and the hypothesis performed at QC, they are not able to identify what would be the source and how it is going to be, increase in this particular batch. Then R&D is supporting to do the identification of this impurity, and we perform the, mass technique and mass analysis is this particular samples, and we identify this particular impurity mass would be 509, and based on the 509 information, we suspect the and we predict the impurity, what kind of impurity would be available. And the database, the literature is it clearly indicated that if in the formulation going to be used for sucrose or fructose, then maybe adduct formation would be available. Not only in the sulflexin, any other like oseltamivir and so many molecule where the adduct formation is happening. But in this particular product, there is no informations are available in the USP as well as the other, data domain. So we predict it and we synthesize this impurity using the Maillard reaction, and we confirm that yes, sulflexin adduct is going to be formed this particular unknown impurity, and we confirm it spiking in doing the validation activity. But once we perform the validation, so the RF is coming more than 1, it means around 1.8 or 2. So the impurity limit become double. So the impurity limit as per ICH should be around zero point 0.4 and zero point 0.5, but the value is coming more than 0.5. So we have to go on for toxicological study, predicting that okay, 1%, 2%, 3% we have to do the toxicological study on the rat, and we have gone through and we completed the 90 days toxicological study, and we confirmed that there is no adverse reaction for this particular impurity in the sulflexin or powder for suspension. Then we perform the validation using the 3% of the specification limit, and informed to the agency, submitted the complete documentation, the stability data as well as the validation and toxicological report, and the regulatory variation has been filed. And once the agency is going to be approve this application, then we are going to continue for the manufacturing and supply the product to the market. Another example for this case study 3 is the combination product of doxycycline and pyridoxine hydrochloride in drug product, and it was observed that there is a unknown impurity This unknown impurity is coming more than 0.2%. It is a 0.25%, and RRT is 0.6. And we identify this unknown impurity mass has been done, and the mass is coming around 320. And based on this mass information, we gone for prediction of this structure. Then we draw the structure, what kind of impurity would be there. So using the hypothesis analysis, using the investigation, we come to know that the pyridine would be uh, the right molecule which is going to be degrade, and in presence of heat, and this particular impurity is going to be formed where the mass is matching in the particular structure. So this pyridoxine impurity we synthesize and updated in the specification and informed to the regulatory agency, and regulatory agency is given the acceptance to include in the specification, and based on their acceptance and approval, we have gone through the commercialization and batch analysis uh, using this particular known impurity in the commercial batches. Another example of case study, this rasaglin tablet. So in rasaglin also, during analysis, there is unknown impurity observed, and after complete investigation and hypothesis performed at quality control, they could not able to identify the actual root cause, and so we took up, they took a help from R&D, and we have supported with the help of mass spectroscopy, especially develop the method and identify the particular mask, what kind of mask would be available in this is recycling, how this impurities are coming and what would be the source of this impurity. So once we check the mass and completely based on the database, based on the information, this mass is not matching with the recycling. What would be the root cause of this mass? Then we have gone through the complete information from the analysis and quality control. Okay, what kind of analysis is going to be take place when this recycling analysis is going on. So we come to know that another product is going on for analysis and quality control, and due to cross contamination, this impurity may come up in the from the flask itself, and the flask was not completely clean as per the procedure, and that flask has been used during RS analysis, and this cross contamination is happening, and we confirm it that would be the different product, and the cross contamination is giving in this particular batch analysis, and according to the complete investigation, and the report has been submitted to the quality control, and accordingly they have made the kappa to issue the glassware to the scientist accordingly. So these kind of based on this information, based on this hypothesis and the complete investigation, from taking the from the mass spectroscopy, and appropriate kappa has been established. Now coming to the challenge of this NTA and SSA. Of course, this NTA is generation the vast amount of data, making difficult to process, filter, identify the relevant features. Unknown impurity identification is also very, very challenging. The data assessment, evaluation, and method development, validation, information to the agency is also very, very challenging task uh, to identify this unknown impurities. And the matrix effect is also, playing a big role for formulations and all. And sometimes this, mass information and the mass technique may not work if we are going to use the complex formulation where the polymorph, like a polymer is going to be used in the formulation. So we are getting a lot of challenges to get the proper mass in this particular polymer, samples if it is using in the formulation. And the lack of standardization method, as I said, that in industries, if you want to, identify the unknown impurity, there is no database is available from the, any specific regulatory guideline on any pharmacopoeia methods are available for to identify this NTA. So this is the biggest challenge. Every company, every person is going to do the proper identification using their methodology and justify to the agency. So this is also, big challenge to most of the industries, analytical person. And interpretation of the result is also very, very, challenging, because depend upon the source of contamination, source of impurity, from where it is coming, and different kind of metrics is also, challenging in this. And skill gap, yes, the high-end instrument handling of this mass spectroscopy is also very, very, challenging, because most of the people should be, properly skilled for handling those instrument, and do the proper development and validation activities and all. And the risk mitigation, yes, of course, this unknown impurity identification is helping a lot using this mass technique, and do the complete characterization, because apart from mass, we have to use the NMR, we have to use the IR. So sophisticated instruments are available, and these instruments are going to help us to do the proper, characterization, structure illustration report, which can be justified to the, regulatory audits, and which can be justified to the FDA to give a, approval for the particular impurities. And once we are going to, generate the data, most of the company having a LCMS and triple quad also, but in LCMS triple quad don't have a library, but GCMS, of course, libraries are available. Then we have to do the, investigation and make it known the impurities, and the library can be, generated. So these, information in future is going to help us not to do the complete investigation. If any unknown impurities are coming up, we have to run the mass and check that, mass is matching in the library, is helping us to predict the structure and inform to the, quality person, yes, these impurities are coming up, and this would be the root cause of this particular impurity. So this database is helping us to take a decision faster. And there's a, structure prediction is also available in the de novo, and that is also going to help us to predict the structure for a known impurity nowadays. Now, coming to the NTA SSA, as I said, that is a critical tool for prediction of quality assurance, because in quality control, this unknown impurity need to be, properly identify. The root cause investigation has to be done, and we need to ensure that this unknown impurity should not be, coming as per the specification limit. And detection of this unknown impurity now becomes very, very helpful using this fast technique. And this detection in different sources from the formulation or the packaging material can be easily identified using this mass technique. When implemented like these kind of, database and submitted to the agency, then regulatory agency is going to be approved after assessing the proper safety, risk of the human patient. And nowadays, like software is also, helping us to do the proper assessment of multivariance statistical, data, and using the different technique, especially mass, more become more user friendly to the analyst to taking a decision, and supporting to take the safety risk of the specification and the quality of the product. And finally, the quality assurance is non-targeted method is being discussed, but has not yet regulated. As I said that yes, every, company, every analyst is going to be develop the method, validate the method, and submit it to the agency. But the database in the regulatory body or maybe any pharmacopoeia is not yet, established. Another, like, advantage of this mass technique, the yes, different mass is available where we can use it to versatile and sensitivity check in the different metrics to perform the analysis, to check the impurities, what kind of impurity would be available. The LCMS would be really helping us to go with the PPB label, to develop the method in the sensitive technique, especially in the mass. So these are the sensitive technique, really crucial, but is really helping us to identify this unknown impurity at the detection level in the drug substance as well as the drug product or any kind of raw material as well. And the identification and characterization is also very, very important to make it known of this impurity from like, NTA to SSA or target compound. Once we identify the target compound helping us this mass spectroscopy, then other software and other advanced technique is helping us to illustrate the structure and inform to the agency to get the proper approval for this particular known impurity. And this application across the field is definitely this mass is nowadays is going to use for not only in the pharmaceutical company, it is going to use for the most of the field like environmental science, food analysis, biometrics analysis, forensic and protein estimations and all. So these are the high instrument and software is going to help us across the various field. And another advantage for LCMS is very, very, comprehensive information providing by, doing the proper mass assessment and impurity identification is compared to the liquid chromatography and gas chromatography. And the combination of this chromatography and mass spectroscopy nowadays, the people are using the coupled with the LC and GCMS and the routine analysis to save the time to if any unknown impurities are coming up, so they are going to check with the LCMS as well as the mass. And the mass is nowadays available in the most of the quality control, and they are going to get the information very faster and get the information uh, predicting the structure and arranging the impurity and completing their hypothesis investigation, and instead of like taking more time. So that's all from my side.
All right. Thank you so much, Dr. Manish, for that lovely session. And I'm sure this was very helpful in understanding the analytical techniques for the non-targeted analysis and suspect screening. We will now move on to the question and answers session. So the first question we have is from Dharmendra Tiwari, Medley Pharma. He asks, what are the regulatory expectations for method verification as per USP 1226? For which type of test method verification will be required? That's number one. Then we have another question in part.
Correct. So thank you so much, Dharmendra. Is a very important questions for USP where we are going to do the method validation activity. So as per USP general chapter, they are clearly defined that we have to do the couple of parameters. So I can define that the force degradation is not required if the monograph is available in the USP. Because in the USP monograph is available, then the USP body is going to do the proper validation, complete validation, including force degradations and all, and then only they're going to publish the monograph. If as per USP general chapter, if the monograph is available, then we can go ahead with the partial validation activity, and it is going to be accepted by FDA.
Okay, before you take the next question, I just wanted to highlight that this particular question is in two parts. There is part one and part two because there is a word limit, and this is why the questions will be put in two parts. So I will show you both the parts of the questions,
Yeah,
and
please,
Dheeraj,
please.
you can moderate it.
Yeah.
Okay. So let's have the next question on screen. All right. Now he mentions when an impurity is well within limits during initial analysis, but co-eludes or is not well separated from a closely eluting peak, close RRT during stability studies.
So you see, first we have to identify whether this closely eluting peak is known impurities or unknown impurity. So first we need to understand whether, so there is the two case. These two closely unknown impurity would be there. Then we have to separate and what would be the limit or what would be the percentage is coming during the stability. That is also very, very important. If let's say in the finished product as per MDT, the limit would be 0.2% and our closely eluted impurities are coming around, more than 0.15, then it is going to be risk at what station these impurities are coming up. And we have to extrapolate and identify this unknown impurity. Okay, and first we have to separate out this impurity. Nowadays, regulatory body is going to ask that each and every impurity should be separated properly to get the proper result or accurate result. If it is the one known impurity and another unknown impurity, then the prediction during analysis may be false. This known impurity may become an unknown impurity, or maybe unknown impurity may become a known impurity. So there is a always confusion during assessment and reporting of the result during QC analysis. So better to go with the proper separation and report the result accordingly.
Thank you. Thank you very much, Manish. And in the continuation, because previously he mentioned about the scenario, and in that particular scenario, he is asking his question is that it becomes a critical issue. This situation can lead to a underestimation of impurities or masking of
Yes, it is a critical issue,
degradants.
and
Yes,
that's why I'm saying yes, it is a critical issue,
yes. So you're
and
looking forward to handle
agency
it.
may ask or during audit also. Yes, it is a critical issue.
Yes, and so the question is, how will it be handled?
Yeah, so that's one thing. We need to understand that what percentage is coming and the closely eluted peak, the percentage, let's say the known impurity limit is a 0.2%, then we have to assess the data. If the data is well within the limit, let's say, less than 0.1%, and these are the unknown impurities not going to be increased during stability analysis, then we can able to justify to the regulatory agency. And if it is going to be, increased during the stability and we observe the trend analysis increasing the impurity, then it is going to be a risk for the failure of the product during stability analysis.
Thank you very much. And the next question we have is from Shikha. She's talking about for Europe market, is it not required to perform method validation for EP pharmacopoeia methods?
Yes, actually in Europe market, usually in the monograph, if the monograph is available in the EP, European Pharmacopoeia, then the CEP got approved. So they always go for the complete, validation for this finished product, and they required a CUA, only certificate of analysis. So the suitability information can be, yeah, the suitability and the CUA would be suffice the purpose, but as per the quality control department, we have to ensure to do the complete, validation and method transfer activity using the different parameters.
Okay, I am talking on the mute. Okay, thank you very much. And
Yeah, it is
again,
not
but
required the fourth. Yeah, yes, it is not required the fourth degradation.
okay, it is not required. And then we have question from Parminder. Hi, can add from my end for the response for this specific query for close eluting peak handling strategy. Uh,
Yeah,
okay. All right, Praminder. So right after the session, I will certainly take you and you can add pointers on that as well. So sounds like a very good plan, and you can build on this particular approach. And we have more questions for you. We have Smita again, and here's the question: what will be the challenges to convert known unknown to known known? As in many monographs have these known unknown, and why limit of unspecified and not applied to known unknown?
OS, then we have to go for this, make it
Manish,
known.
can you hear me?
Yeah, yeah, I can. Hello, am I audible?
Yes, yes, you're audible now. You're audible.
Okay, okay. So that's why I'm saying this all depend upon the data assessment. During analysis, we have to check whether the trend is the particular unspecified impurities are well within the limit or whether it is going to be increased during stability. Then accordingly, we have to take a call, make it known.
So this is the next question that we have, Manish. We have performed mass analysis and molecular weight for unknown impurity is identified. However, we cannot predict structure based on the literature. So is there any other technique to predict structure?
Yeah, in silico and different softwares are available. Then we have to, take a help from the experts from the chemistry background and predict the structure. Yeah, definitely the different structure can be identified with the help of mass, and we need to synthesize this particular impurity. Once we identify the, probable closely, predict structure.
All right, thank you so much for that. And now we have our next question. It mentions what are the acceptance criteria for the relative retention time? Is it based on monographs? And if you can elaborate on that.
You see, our relative retention time is there is no criteria, but yes, the peak need to be uh, separated well. So as per the USP, the minimum resolution should be 1.5.
Okay.
Yeah. So RRT accordingly, RRT can be calculated.
All right.
Yeah.
Thank you so much for that.
Yeah, if it is closely diluted, then RRT would be very close. Then during reporting, it is very difficult to identify whether it is a known impurity, whether it is an unknown impurity. So that's the reason the USP and the FDA is always insist to do the proper resolution between two peaks.
All right. Thank you so much for that, Dr. Manish. And that explanation was amazing. And I'm sure we had a lot of insights on this, and everyone, just were in awe of it. So now I would like to thank you once again for it. And if you'd like to conclude, and then we will move on to the next session.
Yeah, thank you so much, Dheeraj. Thank you.